Hi everyone, it is 2:30 Eastern Time, so we are going to get started with another of Edvotek's Biotechnology live streams. Many of you may already know me, but for those who don't yet, my name is Dr. Danielle Snowflack. I am the Senior Director of Education at Edvotek, and I'm so happy to be with you again this month to discuss biotechnology techniques that you can use in the lab, in your classroom lab, in your research lab. Pretty much anywhere you can do science, and that's the beauty of the EDGE, is that it's actually really able to be transported. It's portable, it's compact, and it's an all-in-one system. So I'm really excited to be working through this biotechnology lesson with you, using our new equipment, the Edvotek EDGE, and it's going to be a really fun workshop. Let's get started. And for those of you who it may be your first live stream, I'll tell you a little bit about Edvotek. We are the biotechnology education company. We were founded over 30 years ago by Dr. Jack Trickjian, who was a professor of biochemistry at Georgetown University. And this was a really exciting time for biotechnology. If we think back to the 1980s, the technology was developing at what seems to be lightning speed, with the advent of the polymerase chain reaction or PCR, genetic engineering or recombinant DNA technology. All of these things were being done in the research lab, but little of it was being translated into teaching classrooms. And thus Edvotek was born to really be able to democratize this technology. And we've continued to do that today, where we are working not only with educators, but with biohackers, with outreach spaces, and even with, researchers who are looking for the best value for their equipment, and for the equipment to be rugged and useful and well-designed. And so we are working with researchers, scientists, educators all over the world to help demystify science, to make it accessible, and to foster the next generation of scientists through hands-on learning activities. But before we start talking about the science, I got one more slide for you. Halloween is coming up. It's a favorite holiday of mine. If you're like me and you love Halloween and science, we have got the perfect contest for you. I want to share our Edvotek pumpkin carving contest. So we want to see your science pumpkins. Be sure to tag us on Instagram or Twitter or both. Tag both at Edvotek and use the hashtag EdvotekHalloween before October 31st. We are going to pick our favorite science pumpkins and give away a $100 Edvotek gift card. So again, you've got until the 31st. There's plenty of time to combine that love for science, art, Halloween, pumpkins, and to really have a lot of fun and to share it with us. We can't wait to see what we've done. We've already seen a couple really fantastic pumpkins. So let us get started. We are using the Edvotek EDGE to do electrophoresis, but we are doing this in the context of a sickle cell disease diagnosis kit. And so sickle cell disease, or SCD, is an inherited blood disorder, and it can cause huge problems for individuals who are affected with it. And so the disease is actually caused by small genetic changes in an individual's genome that interfere with the body's ability to form a fully functional hemoglobin protein. And hemoglobin is the protein that transports oxygen around our bodies and delivers it at different locations. And so I kind of think of, our circulatory system as a highway in our body, and the red blood cells are kind of like, 18-wheeler trucks. They're really responsible for picking up the oxygen in our lungs and delivering it all around the body. And that's largely accomplished through the connection of oxygen to hemoglobin. And so changes to the hemoglobin protein are going to affect these red blood cells. In most people, our red blood cells are disc-shaped, flexible, and smooth, and we can see that over in the picture that we see next to me. You can see over the normal red blood cell is round and kind of like a disc shape with an impression in the middle. And this allows it to be flexible, to bend, and to move really through even small blood vessels. In contrast, a A healthy, normal red blood cell is going to last about 6 months in the body, transporting oxygen around before it's recycled into, recycled or broken down. In contrast, when we change the hemoglobin protein in a specific fashion, it causes the body to create some red blood cells that are crescent or sickle shaped. They're going to be stiff when they're in the body, and the outsides are very sticky, which means they're prone to sticking together to one another, which can form clots in the body, which is a very bad thing. And another caveat is that they only last for about 15 days before they are recycled by the body, which is going to be an energy intensive process, constantly needing to break down and to build new red blood cells to transport oxygen around the body. And so in normal red blood, in people who are not affected by SCD, we have these normal red blood cells that are shaped like these discs, and they can flow freely through the blood vessels, and they can even get through the smallest blood vessels, again, because they can bend and flex to move through even the smallest capillaries that we would see. In contrast, the abnormal red blood cells are rigid and they stick to one another, meaning that they are going to they're going to get stuck in these smaller capillaries and in these junctions because they're clumpy and they're stuck and they're rigid. And they can't twist to fit through the different capillaries. And so because they're stuck to each other, because they're rigid, they are going to form blockages in blood vessels, which is going to form clots and other problems. And so that's really the smallest part of this. Because of the nature of red blood cells, because of how necessary they are in the body, how oxygen and blood is important for every system within our body, individuals with sickle cell disease are going to have, problems in most of their body systems, right? If we can't get oxygen to the nerves, right? If we can't get oxygen to the heart, if we form clots in the heart, if we can't get oxygen to our extremities, to our lungs, these are going to cause, health problems. And so this diagram that we have here on the slide is basically demonstrating some of the ways, some of the places where individuals with SCD normally have problems. And I'm going to point out 3 of the most common, but the conditions really run a whole gamut of different symptoms, ranging from chronic tiredness to blood clots and heart attacks. And so some that are very common are this SCD anemia or sickle cell anemia. And this is characterized by low red blood cell counts and low blood oxygen levels. And that's going to come from the abnormal hemoglobin found within the blood cells and the abnormal shape of the cells itself. There's something called a vaso-occlusive crisis. And so this is the clumping of the cells, the clotting of the cells all together, which is going to block the flow of blood to different organs. And this can be very painful. You're cutting off blood to different organs. You can have tissue death, extensive organ damage if you're not treating these conditions early enough. And depending on the patient, these attacks can be very frequent, very severe, and last for days. A third common condition is going to be spleen sequestration crisis. And this is one that actually, if not treated early enough, can be incredibly fatal. And so the spleen is an immune organ, that helps make immune cells, break immune cells down. It's a storage site for some different cells. But it is a place where red blood cells are common. And the buildup of cells in this organ can be problematic because it's not easy necessarily to break them down and recycle them when they start to accumulate. And so when the spleen fills up with red blood cells, this can happen precipitously, resulting in a fast drop in hemoglobin levels and swelling in the spleen. And this actually is going to end up reducing the concentration of the blood, which then can affect the normal flow of water into and out of the circulatory system. And this is a major emergency that, if not treated, can prove fatal. and it can last for up to a day. And so, individuals who are in this crisis may need blood transfusions and other treatments in order to stay alive. While this is a chronic condition and it is a condition that individuals can live with, and many do have long and productive, healthy lives, there are serious complications that individuals have to negotiate every day when they have sickle cell disease. And so if we look at the population, sickle cell disease is going to affect people, over 5 million people worldwide. And while the prevalence is largely in sub-Saharan Africa, the movement of African populations around the world has caused an increase in SCD prevalence worldwide. Today, again, we see about 5 million cases. And I do want to point out that it's important to mention that being a carrier of SCD, meaning you have one compromised copy of the gene, is actually protective in areas of the world where malaria is common. And so it's possible that it evolved as a mechanism through which individuals have resistance to malaria in the absence of medications, which is likely why it originated in an area with a higher incidence of malaria. And there's some interesting evidence that is starting to come out that this sickle cell protein in low levels can actually prevent the malaria parasite from hijacking red blood cells to reproduce. And so there's a lot of really interesting research here, and we're still learning so much about the disease and how it affects our bodies. And we're going to talk about some exciting biotechnologies that are being used to potentially cure sickle cell disease in individuals who are affected. So because SCD is a genetic disease, biotechnology can be used to identify and treat the disease. So today we are going to perform experiments that model what clinicians might do when they are performing prenatal testing for SCD and other hemoglobinopathies, so other diseases of the hemoglobin of the red blood cells and of the blood. And so we are going to use our new Edvotek EDGE, which is exciting. It's our new electrophoresis chamber, and we are going to be using that to perform electrophoresis of kit 116, which is our sickle cell gene detection kit. And we're also going to be talking about a complementary kit, which can be paired with the sickle cell electrophoresis to kind of create a really nice module. And this is our Learn to Code module, which focuses on using the programming language Python for detecting disease. And so we are recording this presentation as always. And again, if you'd like to be notified, when we make the slides available, and if you'd like a professional development certificate, please fill out the form that Maria has linked in our chat window. And we will send that to you next week. For those of you who are here live, again, be sure to fill out that form because I will send you, if you'd like a professional development certificate, but we will only leave the form open until 4:30 Eastern time because we're trying to get people who did tune in live. Let's get started. Let's get started with our electrophoresis experiment. As always, if you have any questions, please put them in the chat box. We'd love to hear about you if you've done this experiment. If you're planning to do this experiment, want to hear what you're doing in biotechnology, or if you want to tell us about your pumpkins, tell us about your science pumpkins. We love to hear that too. So let's get started. So here is a quick summary of electrophoresis. I'm going to go through it quickly since we do offer many videos about electrophoresis, including longer live streams on our YouTube channel. If you have questions, again, put it in that form. I'm happy to answer them. But electrophoresis is the technique that we're going to be using to analyze different banding patterns in people with different, who may or may not have SCD. And we're going to analyze their DNA, which will tell us about their genetics. And so agarose gel electrophoresis is a technology that allows us to use a porous gel matrix and electricity to separate DNA molecules by their size. And so this is a slide, is an overview of what the electrophoresis process would look like. And so we cast our gels, we submerge them in a buffer, and then we load our samples into those wells. We attach a safety cover, our leads, and turn on a power supply. After about 15 minutes or so, we can check on our results. I'm actually going to leave the light on the entire time. So with our Edvotek Edge, the great thing is that we can separate the DNA and watch it separate at the same time, which I think is super amazing. And so, we'll give the gel 15-20 minutes to run. And when we're done, we will analyze our results together. So what do we need to perform electrophoresis? We are going to be using our Edge, and I'm going to switch over to my science view so that we can start doing our electrophoresis experiments. First things first, safety first. I always want to put gloves on. Again, SYBR safe is generally considered to be safe. Agarose, electrophoresis buffer, nothing that we're using is going to be toxic or hazardous, but safety comes first. We always want to protect ourselves. So I am wearing gloves. As I always like to say, my lab safety rules are don't eat it, don't drink it, don't touch it with your hands. Always wear gloves. And so, this is one of the ways that we can protect ourselves. So we are doing electrophoresis today. Electrophoresis is a very versatile technique. We are going to be using our samples that come in kit 116. They are going to come in our quick strip format. One thing I love is that the bags already have the label on it telling you how to store them. But we are doing kit 116. This is our electrophoresis of sickle cell DNA. And so I'm going to pull out our quick strips. And quick strips, are fantastic. They're fantastic. They are pre-aliquoted samples. So basically everything you need to perform the experiment in terms of the DNA is already in the sample. We have our DNA, we have our loading buffer. That loading buffer is important because it's got a buffer in it there that's going to keep the DNA at a happy pH, meaning it'll be stable and ready to go in our electrophoresis experiments. It's also got a tracking guide, which will allow us to see how far our DNA moves through our gel. And the nice thing is that the samples are pre-aliquoted, and that means that I just take one of these strips. We're going to give one of these to each of our groups, and that's all the prep you need to do on this end. So super easy way to do electrophoresis. So electrophoresis itself is super versatile. It can be used to separate a variety of different biomolecules, DNA, RNA, protein. Again, we're doing DNA today. We're doing it in our Edge. Let me pull up the camera a little bit. Oh, let's not look at my messy floor, but let's see more of the Edge. You can see there is our timer. Here is our orange filter. And so we are doing agarose gel electrophoresis today. Agarose is our matrix for separation. I call it my science jello. And so this agarose, which I have cast in our casting system, this agarose is going to act like a strainer or molecular sieve, which is going to allow us to separate our different DNA molecules by size. And that's important for the results of our experiment. So I've actually already added SYBR Safe to our gel. SYBR Safe is an important addition to our electrophoresis experiments. It is going to allow us to visualize our DNA. Our DNA is clear and colorless, and so we need a way to see it. And so this electrophoresis system is actually optimized to use with SYBR Safe because of the blue light system, which excites the SYBR Safe and allows us to visualize the DNA. The electrophoresis buffer, which I'm going to add, I have pre-diluted. It's a mixture of buffers and salts. And again, there are going to be two reasons why we use this TAE electrophoresis buffer. Some kits use TBE. It's a different system, but the basic biology is the same. There are two reasons to use buffer instead of water. First, we are using electricity to separate the molecules. Water is a super poor conductor of electricity, and the salts in the buffer in here mix with the water, which allows our current to flow. And next, again, because we are using biological samples, we got to keep them at the correct pH for them to be stable and also to have a charge to be separated through our gel. And so, we include our buffer as a concentrate with every one of our electrophoresis kits. And that's basically going to make your prep easier because you receive a small box with everything. I'm going to turn this over so you don't see my address, which is taped on the back. You will receive a small box. It's almost like the size of like a hair product or a beauty product. You receive this small box, and you dilute it. You prepare everything at home. There's not huge volumes, huge storage concerns. I have my gel in here in our electrophoresis chamber. Today I am using the Edvotek EDGE, which is our fantastic new all-in-one unit. This unit is going to combine the electrophoresis chamber, the power supply, and the visualization system all into one compact, easy-to-use unit. If you have any questions specifically about the EDGE, definitely let me know. I'm happy to answer them, and I'll be showing this. I'll be showing the EDGE off all day in this. Well, for the next 45 minutes in this workshop. There are many advantages to our system over others, but the one that I think is really important is that our wells are the same size as our M12 wells. So they're larger than other all-in-one systems, which makes our system really easy to load as compared to some of the others, which have smaller wells, which maybe they can only accommodate 10 microliters or so of sample. You can load between 20 and 35 microliters of sample into these wells, no problem. And they're larger, they're easier targets for your students to find, and basically to load the gel. And you can see, I have loaded many gels, both on YouTube and in person, but even under this most difficult of circumstances, it's pretty easy for me to get the samples in here. I'm loading our samples with an adjustable volume micropipette. This is a precision tool that's going to allow us to measure and move very small volumes of liquid. And you can see when I'm using the quick strip, I take my pipette tip, micropipette tip, I puncture the foil, and then I draw the sample up. Get that last sample in there. And then we've got one more sample to load. And so while you're looking at this, what you should see is that our electrophoresis chamber is going to have electrodes wired into either end of it. The leads are plugged into the integrated power supply, and that's going to actually generate the current that we need to perform electrophoresis, so that we need to push our DNA samples through the gel. So I have the gel loaded. That was easy enough to do. What we do to run the EDGE is we're going to put our orange cover down. One advantage to our system is that it can only run with a cover on it. You can't turn it on without the cover, or because it will not allow you to turn the current on. This basically means that your students can't get into it while it's running and potentially, get a little jolt. So I'm going to run it here. I'll show you what I'm programming, 150 volts. I'm going to set it for 30 minutes. We shouldn't need that long, but I just want to keep it running while I'm talking. In case I talk a little long, which you all know I sometimes do. We'll just make sure the gel is running the entire time. And I'm going to hit start, and we're going to go. I'm going to turn the light on as well. And I'm also going to turn off my extra light so that we can see our bands as they're separating in real time. You can see this even in a well-lit room, but it's nice to draw the lights down a little bit, especially to allow my camera to really see what's going on. Okay. If you have more questions about electrophoresis, make sure to put it in the form that Maria has, posted here, or check out one of our other electrophoresis videos on YouTube. All right, so let's talk more about sickle cell disease. And again, sickle cell disease is going to be a disease of our hemoglobin. So let's focus on what this molecule actually is. And so hemoglobin is actually not a single molecule, it is a heterotetramer. So if we break that word down, hetero is going to mean different. Tetra is 4, and in biology, a mer, M-E-R, is a unit. So when we say something is a polymer, it means that we are linking many units. A tetramer specifically is built from 4 units. And if we have a heterotetramer, that means that those 4 units are different. And so in a typical molecule of hemoglobin, we have 2 subunits, 2 each, in our molecule. And so we can see our alpha hemoglobin here is in red, and our beta hemoglobin is in blue. And we can see that we have 2 of each of those in our hemoglobin molecule. We have 2 of the, and so each monomer has a iron-containing heme group. Let me actually bring this camera down a little bit so we can really start to see those bands separating. You should already be able to see the little bands separating in the wells. This just gives us a zoomed in version so you can see it better. All right, so again, hemoglobin is made of 4 monomers, 2 of which are alpha hemoglobin, 2 of which are beta. Each monomer has an iron-containing heme group, which is the group that's attached, which is responsible for binding and transporting oxygen through the body. In healthy cells, hemoglobin exists as free tetramers within the red blood cell. And I forgot to advance. Here is our heme molecule. That's a prosthetic group that is going to be bound with each of the hemoglobin monomers. And you can see in the middle, that's where the iron molecule is, and that iron molecule is important for the ability of hemoglobin to bind with oxygen. So again, hemoglobin is tetrameric, but each of those tetramers is kind of free to be with itself in the red blood cell as the red blood cell circulates through the body. However, when we make changes to the protein sequence of hemoglobin, problems can occur. So researchers have actually identified more than 400 mutations in the beta hemoglobin gene that can cause SCD. The most common genetic change is a single point mutation from A to T, and we can see it here. If we look at figure 2, we can see that glutamic acid, which is a GAG, and if we look at the mutant form of our sickle cell beta hemoglobin, we can see in the same place in the mutant we have a GTG, which is going to change the amino acid sequence of the protein. Essentially, this single base pair is going to change the codon, which then swaps out glutamic acid for valine. And so the reason that this is so detrimental is because these 2 amino acids have different properties from one another. If we look at the structure of them, we can see that the change from the negatively charged glutamic acid to the hydrophobic valine actually is going to influence the protein structure. As opposed to the wild type hemoglobin, which is going to exist as a free tetramer within the red blood cell, the mutated hemoglobin forms long protein polymers that stretch the red blood cells. And they are rigid, and this is part of the reason why we get these distorted sickle shapes, but we also get rigidity, right? So in a wild type red blood cell, they're soft, they're flexible because these tetramers of hemoglobin are free to be within the cell. If we form them into like these long thread-like projections, it's kind of going to be like a tent pole where it pushes the shape of the fabric, out in a way that creates rigidity. And so that rigidity is going to create the distorted sickle shape characteristic of the disease. So because this is a genetic disease, how can couples who are considering children who may be in a risk group for this disease, how could they determine what their genotype is? And so routine prenatal testing analyzes the patient's blood for hemoglobinopathies or diseases of the hemoglobin and in red blood cells. The first test that a clinician may do when a pregnant person comes to a clinic is to look at the level of red blood cells in the blood sample. If the sample does not match the expected range, then further analysis at that point is going to be performed. Hemoglobin electrophoresis is another test that's used to evaluate the parent's blood, and that is exactly what it sounds like. The parent's blood is analyzed using electrophoresis to determine the amount of each of the protein subunits within hemoglobin. So remember, in normal wild-type hemoglobin, we have our HbA, our HbB, and we have 2 of each of those. Should one of those subunits be compromised, we would be able to see it run differently on our electrophoresis gel. This would allow us to determine whether there are different forms of hemoglobin present within a patient sample which might indicate disease. Finally, there is genetic testing. For example, as we can kind of see here in these schematics, many SCD alleles are recessive, which means that one wild type gene can hide the disease. So If we're talking about recessive genes, you're going to need 2 copies, both inherited from your mother and your father, to have a particular phenotype. And so we can actually use biotechnology to read the exact sequence of A's, T's, G's, and C's within a patient's genome, and then to identify changes within. If any of the tests from the pregnant parent come back positive, the other parent is tested to determine the likelihood of genetic disease. So let's talk about, since we're doing an electrophoresis experiment, we're talking about DNA, and we're talking about the genetic changes. Let's talk about what the genetic markers that we might look like would be. And so some of these common landmarks are going to be single nucleotide polymorphisms, or SNPs. If you've followed any of my DNA fingerprinting workshops, you should know all about SNPs at this point, but I'm still going to do a quick review for those of you who may not have joined me for those workshops. So a SNP, or a single nucleotide polymorphism, is a place within the genome where the DNA base pairs at a specific location shows variability. So this is where, if we're talking about sickle cell disease, this is where one individual has an A and one individual has a T, and the A allele is our wild type allele and the T allele is our recessive disease allele. Now, some of these DNA sequence differences are considered to be restriction fragment length polymorphisms, or RFLPs. So here's another term. What does this term mean? So let's step back a second and let's digest that term, restriction fragment length polymorphism. So if we're thinking about restriction fragments, they are generated by a restriction endonuclease, or a restriction enzyme, which is a highly specific enzyme that cuts DNA into fragments based on its sequence. It's like a molecular scissor, which can only cut at specific locations, and the sequence has to match exactly. For example, as we can see on this slide, the restriction enzyme EcoR1 cuts DNA with the sequence GAATTC. Let's say that that second A is where a SNP can occur. It can be an A or a T. And the enzyme will only cut when the DNA has an A in that location, because it relies exactly on that GAATTC sequence. If we had GATTTC, then the enzyme couldn't recognize the DNA and it wouldn't be able to cut the backbone. So if we look here at this schematic, we have a segment of DNA between 2 black arrows. There are 2 potential RFLPs here, one indicated by the yellow arrow in DNA sample A and one by the red arrow in DNA sample B. If we were to just isolate this DNA and run it on a gel, the 2 would look the same. However, when we cut with EcoR1, we see different results. For example, in sample A, the cut site is closer to the middle, so our DNA fragments are closer in size. In sample B, the cut site is closer to the end, so the 2 fragments are more different in size. The differences in this pattern allows us to distinguish between people and to identify disease states. So before I talk about the next slide, I just want to say definitely take a look. You can see our DNA fragments are separating, and this has been, about 10 minutes right now, and you can see we're already seeing our banding patterns, and we can already do some analysis. We're going to keep letting it run while I'm talking, but the beauty of the EDGE is that we can watch our DNA separate in real time while we're talking about our science. Okay. So let's talk about how we do RFLP analysis to analyze our beta hemoglobin gene. So you may see that as HBB on this slide, which stands for hemoglobin beta. And so we have been, due to biotechnology, we have been able to sequence the entire gene, and we have recognized that the restriction enzyme MST2 recognizes the sequence CTTNAGG, where N can be any nucleotide. In the normal beta globin gene, the nucleotide sequence is here on the screen, so you can see it's CTTGAGGAG. And the green site that I've highlighted there is recognized by MST2. And so you can see, because it's got CTT, N, which is any nucleotide, which is represented by a G here, and then we have an AGG. Now, when we convert our DNA, and we have this point mutation from an A that converts the A to the T, it changes the sequence of that restriction site to CCT gene. And so this is not able to be recognized by MST2 because it takes that A, it changes it to a T, our enzyme cannot cut. And so therefore, when we mix a patient sample with MST2, the enzyme will cut the normal gene into two pieces, but it cannot cut the mutated gene. And so again, if we think about human genetics, each individual has two chromosomes, one from the father, one from the mother. And those, meaning that when we digest our DNA, there are three possible combinations. If you're homozygous for either allele, meaning that you have two copies of it, you'll see what either the leftmost or the rightmost images. So you'll see both pieces of DNA are cut the same way by the enzyme. In a person who's heterozygous, meaning that they have one copy of each allele, we will see three bands, meaning we can see both alleles present on the gel. And that's what makes flip analysis really powerful, is that by just digesting one small piece of DNA with one enzyme, we're able to figure out the genotype of an individual at a specific location. And so, this is one way to test, but, many times in research, the entire genome of these individuals will be sequenced to look for different mutations. And so once we have this genetic information from thousands and thousands of patients, what do we actually do with it? So researchers can build programs that manage and analyze DNA sequence data to identify mutations. This requires massive systems to organize and analyze the vast amounts of data being generated. So as a result, scientists have begun to employ computer technologies for storing and processing biological data. Consequently, the interdisciplinary field of bioinformatics evolved, which integrates computer science, biology, and information technology into one discipline that really can tell us a lot about what the genome looks like and what it's doing. And so, while we don't have the time to completely run through this programming kit, it's really a great one to have students do as an independent exploration. They could do it at home, they could do it in class, but programming is such an important STEM skill, and exposing students to programming early increases their interest and ability. But I don't know about you, I'm not a programmer. I have a little bit of background in it from when I was in high school, which was longer ago than I care to admit. But I do think that programming is an important skill for individuals to learn. And so this kit is perfect because it allows you to introduce your students to programming and to really start to try it. But even if you don't have a coding background, it gives you the resources you need to get students trying to program. And so this experiment is going to walk you and your students through what it takes to create and search a database using DNA sequence data from the HBB gene. This exercise demonstrates how bioinformaticians process gene data, how they make sense of gene behavior, and what happens genotypically when a mutation occurs in a DNA sequence. We'll be using Python to quickly analyze large segments of DNA to determine where there might be a nucleotide change. Okay, so we do have a question. What do we do with the M36 if you do purchase the EDGE? And so one thing I would say is that, every awesome piece of equipment does have a caveat. One thing is that I would if you do any dye electrophoresis experiments, I definitely would continue using the M36 for that. And so again, with the dye electrophoresis experiment, we have lots of different colors, right? And the EDGE has an orange filter. And so that is going to affect the way you can actually visualize some of those bands when the experiment is being performed, right? So the beauty of the dye electrophoresis experiments is, again, you can watch the DNA as it's separating through. You can watch the dyes as it's separating through. Throwing that yellow filter on, the orange filter on, and having the blue light, does affect the way that those experiments are run. It's not to say we can't do dye electrophoresis experiments in the EDGE, and it's not to say that, the DNA experiments that you're already doing need to be done in the EDGE. But it is one of the caveats. Having the M36 and the power source, it's, a small package to store in that it's 2 units in opposed to an entire classroom where you would need multiple edges. The edge itself does not accommodate multiple gels, but the casting tray is designed such that it has, as with our regular trays, it has notches where you can have 2 combs placed into the edge, so you can technically get 2 groups in the unit per edge. We do kind of call it an individual electrophoresis experiment, but it doesn't need to be. You can run 2 student groups per edge if you need it to. And then one place where you don't get a bottleneck with this is you don't get a bottleneck in terms of loading, and then you don't get a bottleneck in terms of visualization because each unit has its own visualization system. And so I think, as with any lab equipment, you need to evaluate the needs for your classroom. And the M36 and the power supply are amazing. And if that's what works best in your classroom, that's an option. But maybe if you are working in a classroom, you're working in an outreach situation where you send one of these units to a classroom, you may only need to send one of these for a demonstration, and then you're not sending a separate power supply and a separate electrophoresis chamber and a separate visualization system. So it really is just based on what you like best, what works for your classroom. All biotechnology is awesome biotechnology. It's just that, everybody has their favorite operate. Yeah, it's like Mac versus PC, people will fight you over which is better, but really the best one is the one that gets the job done for you. So I hope that, one thing I do love about the edge is that the wells in the edge are the same size. You get the same casting tray and combs with the edge that you do with the M36 and our other electrophoresis chambers. The only real difference is the edge chamber, the edge casting tray does have these little fins on it, which keep it better anchored. But most of the equipment, most of the casting stuff is actually swappable between systems. And so it could be that this is an additional unit that you may use if you have some research students who are doing different projects and we don't want to pull out the entire M36 for that. So hopefully that helped answer your question. If not, please put more info into the chat and I will answer. But I do want to talk a little bit more about sickle cell disease. It's a disease that is chronic in that most people are kind of living with it. There are ways to manage the symptoms, both manage the symptoms and avoid specific triggers, which I've listed here. Dehydration, illness, and even strenuous exercise can trigger SCD symptoms. There's some evidence that supplements like calcium, vitamin D, and L-glutamine can help prevent SCR attacks, but none of these are necessarily curative. So these are just ways that individuals can kind of keep themselves healthy so as to not have problems and not have these SCD attacks. But today, medical researchers are working to find a cure for the disease. In the most severe cases, bone transplantation is an option. So this is a very, very time-intensive, dangerous procedure, which requires a closely matched marrow donor, several rounds of chemotherapy, and years of taking immunosuppressant drugs. Did I just hear something click? No, it's still running. Okay. Today, and what I think is an exciting technology, researchers are actually investigating and testing another potential cure, which is the use of gene therapy using biotechnologies like CRISPR. And so I did cover CRISPR in a previous live stream that you may have joined me for. That is also available on our YouTube page. But CRISPR-based gene technologies are being developed to treat diseases that affect hemoglobin. So not just SCD, but also beta thalassemia, and other diseases that affect the structure and function of hemoglobin, but also affect the shape of red blood cells. And all of these are going to result in problems with oxygen exchange. But humans actually have several different hemoglobins, one that is expressed in fetuses and babies and the other in adults. the production of fetal hemoglobin turns off around age 2 when hemoglobin A production increases. So scientists realized that that SCD and beta thalassemia, like these hemoglobinopathies, are actually milder in people who still produced fetal hemoglobin. And so these people actually, when they went to the genomic data and they did the bioinformatic analysis, they noticed that these people had changes in the promoter sequence of the fetal hemoglobin gene, meaning that that fetal hemoglobin was still being produced. And so this was a perfect target for CRISPR. It was a gene inside the body that could lessen the symptoms of sickle cell disease and other hemoglobinopathies. And so why not target it to keep that gene turned on? And so they're doing this using CRISPR, which is a gene editing tool, which you may have heard of in the news. It's recently won the Nobel Prize, and it is an amazing technology. And so the first thing I think is amazing is that this is a technology, a system that evolved in bacteria as a defense against viral attacks. So kind of like a primitive immune system. But through research performed in the last 10 years, scientists have actually been able to use the CRISPR-Cas9 system to genetically modify DNA sequences quickly, cheaply, and precisely. The development of this system as a gene editing tool is one of the most exciting biotechnology breakthroughs of the last decade. The basics of CRISPR were discovered over 30 years ago, starting with the identification and characterization of the system in bacteria. In the early 2000s, 3 research groups began to develop CRISPR into a gene editing tool. They recognized that using the system, we could find any short sequence of DNA and attack it with precision. All 3 of these scientists contributed to the research that made CRISPR a real research tool and not just an interesting phenomenon. Now, there's a lot of science drama surrounding CRISPR and the patent situation, and the awarding of the Nobel Prize, the applications of the technology, the development of the parts, and more. And so I'm not going to cover it here because we're not talking about CRISPR here, but it would be an interesting thing to have your students research and explore as an independent study, or even to provide some students some articles for reading and discussion. So we can take this CRISPR and we can actually use it to edit the promoter of the fetal hemoglobin gene. And the way we did this is to create all the CRISPR reagents in the lab. We then remove bone marrow from the patients, and the patient's bone marrow is then destroyed using radiation. The removed bone marrow is treated with CRISPR to change the sequence of the fetal hemoglobin promoter to turn it on constitutively, meaning that when the bone marrow is inside the body, the individual is producing this fetal hemoglobin. The cells are put back into the bone, which is basically a bone marrow transplant with your own bone marrow. So we get rid of all of the problems with rejection and other immune system problems. And so that's put back into the individual. And this has actually been done in several individuals to date. And of the people who have been treated using CRISPR bone marrow, the individuals have not required blood transfusion since the treatment, but do, of course, still require follow-up. So while we were chatting, our samples have been running through the gel and separating by size. So let's talk about our gels. Let's talk about what's going on in the gel. So what does it mean when we add our samples to the wells and we apply current? In this experiment, we are separating using agarose gel electrophoresis to separate DNA fragments by size. Due to the chemical structure of DNA, that's the sugar phosphate bonds that are holding our nucleotides together, our DNA backbone has a strong negative charge. So in our gel, we locate the wells near the negative electrode end of the gel, which is up here. We load the samples and we turn on the current. Oops, sorry. I was just checking something really quickly. So yeah, we load our samples, we turn on the current. The current then pushes the DNA from the negative side of the gel to the positive side of the gel. Now let's talk a little bit about the gel itself. At first glance, our gel kind of looks like a solid jello. It's thick, it's still soft, but a little bit less jiggly than your dessert. At the molecular level, our gel is full of pores, which are these microscopic channels that go through the gel. And these tunnels, these pores, are going to affect the way the different sized molecules separate into bands. As the current is pushing and pulling the DNA through the gel, the molecules have to find their way through the pores. They may have to twist and turn to get through the channels. Since it's easier for smaller pieces to fit through the channels than for larger ones, the DNA is going to separate into bands by size, with larger molecules near the wells and smaller molecules further down the gel. This means that we are going to form our different bands. Because they are traveling, their molecules are traveling, molecules with different sizes are going to travel through the gel at different speeds. They are going to become separated and form discrete bands within the gel. So if we're looking for an analogy for electrophoresis, and this is one I use a lot, but I think it really helps, we can think about DNA molecules like fish, which are swimming down a river. We have some fish that are large, we have some fish that are small. They are moving downstream with the current. So the current here is like our electrical current. We have rocks in the river, or maybe a fishing net that spans from bank to bank. And that's going to be kind of like our aguros. These obstacles get in the way of the fish as they progress down the stream. The smaller fish are more easily able to navigate through any tight turns and twists or fit through holes in the net. They can do this more easily than the larger fish. This means that the smaller fish would travel downstream more quickly than the larger fish. We can think of the fish as DNA molecules, the current of the stream as our electrical current, and the obstacles as our agarose matrix. So if we look at the results of our DNA electrophoresis experiment, we find smaller bands at the bottom and larger bands at the top. We always run a molecular weight marker with DNA bands of known size so that we can determine the size of our unknown fragments in our experimental samples. But there's one more problem. DNA is clear and colorless. We cannot see these bands by the naked eye. And so we need to visualize it using a stain that sticks to DNA. There are many DNA stains available for use in the classroom lab, but I'm going to talk about CyberSafe, which I've already put in this gel. So CyberSafe is a fluorescent dye that binds to DNA. We add it to the gel and it stains the DNA while the gel is running, which means we don't have to spend a lot of time staining the gel after running it. Each DNA molecule is decorated with the CyberSafe, which we visualize by exciting it with blue light. It's very sensitive because each excited dye molecule can emit a lot of light. And so this is an amplification effect, which allows us to visualize the smallest bands of DNA. Okay, so let's take a look at our results from this experiment. The EDGE just turned off. It's been going for 30 minutes. And what we can see when I have the light on, it's a little bit faint here. And I think, again, because there's ambient light in the background here, and I might be getting a little bit, I don't know if my camera is picking up the bands as well now that they're a little bit further apart. I can see them really well on my gel when I look at them. You can also see them here. You can also hear them here in a flash blue stained gel. And what we can see is that the lanes in the digested DNA in each lane created different patterns based on their sequence. So if we look at let me see if I go a little closer, if I can really pick up those bands better. Excuse the wobble while I focus the camera. Yeah, and so when I focus a little closer, excuse the wobble and the moving. Sometimes it's subtle to pick up these things with your webcam. But in person, I have these beautiful glowing bands, which you can definitely see here. Okay, so in our first lane here, let me see, where's my marker? So my first lane there, this is our sickle cell disease control. You can see our DNA is uncut. Our second is our carrier control, where we can see 3 bands. And our third is our wild type control, where we see 2 bands. And basically, what this shows is that in our sickle cell control, we have basically what we have here is we have a person who has 2 identical copies of the gene, neither of which can be cut with our enzyme. In our wild type control, we have 2 copies, both are digested, meaning they both have the wild type gene sequence. Excuse me. And in our carrier control, We have one copy that can digest and we have one copy that does not. What we can see in our parents in lanes 4 and 6 is we can see that they're homozygous and that we can see all three. Let's see if I can move. Oops. Sorry, let's see if I can move this over so we don't get our little focus square in the way. That's not working any better. Let's see if I go that way. Yeah. So here we can see our last 3 lanes in the gel. We've got 4, 5, and 6. This is parent 1. This is parent 2. They're both carriers. We can see 3 bands. In the middle, we only see 1 band, and that is suggesting that they have 2 copies of the recessive DNA, meaning that they have sickle cell disease. So through this experiment, we did use our all-in-one electrophoresis system. And this system, I think, is really important because it's going to simplify classroom experimentation. As with the question asked before, the most important thing I would say is that your electrophoresis experience may vary. Some people will have equipment, and there's no reason not to do these same experiments with the equipment you already have. The biotechnology setup that best matches your classroom. However, if you're looking for a new option, if you want a portable option, the Edvotek EDGE is a fantastic option to simplify your classroom experimentation because you don't need separate power supplies, electrophoresis chambers, and visualization systems. And so if there's any more questions about the EDGE, put them in the chat now. I'm going to kind of continue to wrap up. And so a lot of what we talked about today was sickle cell disease, which is a genetic condition that is going to affect the structure of a protein called hemoglobin, which is found in our red blood cells. That protein is responsible for moving oxygen through our body. In patients with sickle cell disease, those red blood cells do not work as effectively. They can build up in different areas of the body, and they can really cause harmful symptoms in patients. Patients with SCD can experience severe health crises, including pain, blood clots, fatigue, and more. But because it's a genetic disease, there are lots of ways that we can identify and analyze these problems so that we can best treat them. We talked about looking at the sequence of the gene and using bioinformatics to explore it further. We live in an exciting era for biotechnology, and this is important for our students to know because using the power of a technology called CRISPR, which is a gene editing technology, we have the potential to fix mutated genes and potentially cure disease. And so again, thank you so much for your time. Thank you for joining us. If you would like the slides, again, be sure to complete the link. And I will send them to you, and I will send a professional development certificate as well. We're so happy to be able to deliver this content to you through YouTube, but there are lots of ways to get in touch with us. You can call, you can email, you can go to our website, or you can hit us up on other social media, including Facebook, Twitter, and Instagram. So I actually finished 2 minutes early today, which is amazing because we all know that doesn't happen. But I just want to say thank you for your time. Thank you for joining me today. And if there's any questions, please reach out. Please get in touch with us. We would love to help you optimize your biotechnology lab. So have a fantastic day. Have a very happy Halloween, and I hope to see you here again next month. Have a great day.